Here we are discussing laboratory session 9. The objective is to determine the dose of coagulant to given water sample using jar test. What is coagulants? The purpose of chemical coagulants in water treatment is to destabilize suspended contaminants. If the water is containing some contaminants, the coagulants is used to treat the water. What is destabilization? First, we'll discuss that destabilization is accomplished by addition or chemical coagulants such as aluminium sulfate, or ferric chloride. First, we'll discuss the practical significance of using such coagulants to treat water. This determination is used in water treatment plants to find out the dose of coagulants for the specific water sample in the coagulation process. The equipment that is used in this laboratory test is that turbidimeter, pH meter, sample cell, beakers, wash bottles to wash the beakers, polyplater, and the reagents we will use in this lab experiment would be alum and wastewater. Our sample would be the wastewater that we will treat, so we will use that wastewater. The procedure involved in this lab is that shake the sample and put the sample into the turbidimeter cell, read the initial turbidity from instrument scale, measure the initial pH of scale of water sample. 1 liter water is placed in 5 jars and dosed with different amount of coagulants: 100 mg, 200 mg, 300 mg, 400 mg, and 500 milligrams. The fourth step of the procedure would include the jars are mixed at a speed of 60 to 80 rpm to disperse the chemicals for 1 minute. The samples are then stirred slowly at a speed of 30 rpm for 15 minutes. Allow to settle and wait for 30 minutes. Read the final pH and turbidity and plot one graph that would be dose versus turbidity. Dose would be on x-axis and turbidity on to the y-axis. Here we have an observation table that includes the sample volume. We have same volumes in 5 intervals whose initial pH would be 7.8, and we will measure the initial turbidity from the turbidity meter machine. After that, we'll measure the dose of alum on with the measuring unit of milligram per liter. Then we'll observe the final pH using the pH scale and the final turbidity using the machine. Then we plot graphs, and the graphs would be coagulant dose on the x-axis and turbidity on y-axis. We obtain certain elevation and depreciation that can be observed easily. And on the next graph, we will plot pH on the x-axis and turbidity remaining on the y-axis, and then we can observe a depreciated curve. This depreciation and appreciation will lead us to the practical significance of the lab test, that is how we treat water using such coagulant methods and using alums, and estimating the process will lead us to treat water in certain methods. that should be in a very respective manner. So here we have different techniques to observe, with initial and final reading lead us to final conclusions that will lead us to observe more.